Thanks so much for the introduction and for the invitation to speak here today. So I just started my lab here in July, so I'm excited to tell you a little bit about my previous work and then some of the things that we're getting started on in my own lab. Um, so RNA and protein molecules are central to biology, and they're organized across multiple length scales. So individual molecules fold up into three-dimensional conformations, and these molecules can, for example, transport oxygen, sense and respond to metabolites. Many copies of these molecules come together to form macromolecular complexes, things like the ribosome, which is responsible for translating mRNA into proteins. And then hundreds or thousands of these molecules come together to form higher order structures, things that will show up as puncta in fluorescence microscopy images. I'm showing nuclear speckles here, which are involved in splicing pre-mRNA. So the primary sequences of nucleotides or amino acids of these molecules ultimately are the things that are encoding this organization across multiple length scales. And this organization is then encoding function, for example, sensing and responding to stimuli, catalysis, regulation. And so we'd really like to understand, how do we go from to these functions? Recent about 8 billion protein and RNA sequences that exist in nature that are distinct from each other that we know about right now. And so I think we'd love to understand what all of those sequences do and how they do it. But if we're going to get to that scale, we're going to need really efficient methods for characterizing sequence organization function relationships. So today I'll tell you two short stories focused on different length scales of this problem, going from the individual molecular scale to subcellular organization, and then I'll tell you a little bit about what we're getting started on in my own lab. So first, focusing on the molecular level and focusing in on RNA molecules. So RNA plays a very central role in biology. It's responsible for catalyzing reactions, binding small molecules, synthesizing proteins, splicing pre-mRNA, extending chromosome ends, just to mention a few functions. And for RNA to carry out these functions, it folds up into secondary and tertiary structures. So the secondary structure describes the pattern of base pairing within the molecule. The tertiary structure is the three-dimensional structure that the molecule folds up into. And then these 3D structures very often bind to proteins to form intricate RNA protein complexes, and that's how they may carry out their function. So because structure is so integral to the function of these RNA molecules, we'd really like to be able to experimentally characterize these structures. But it turns out that RNA structures are actually extremely challenging to experimentally characterize. So back when we started this project, we just went and looked at all of the structures that had been solved to date. And we could see that the vast, vast, vast majority of the structures are proteins and a teeny tiny minority are of RNA molecules. And RNA structures are also quite time consuming to solve. Typically will take about 1 to 2 years to solve an individual RNA structure. Around this time, cryo-electron microscopy was increasingly becoming the structure determination method of exponential increase in the number of cryo-EM maps that were being solved. 10,000 cryo-EM maps, exactly one of those at this time was of an RNA structure, which is kind of crazy. So we wondered, why is that? The common wisdom at the time was that RNA molecules are basically just too small and flexible to actually be able to resolve with cryo-EM. So we decided, though, instead of just taking this at face value, let's actually test this assumption. So we put together a benchmark set of RNA molecules that span a range of sizes, a range of different functions. They include ribozymes, riboswitches, computationally designed RNAs. And then we collaborated with a fantastic team, including Wachu and 2 postdocs from his lab, who now run their own labs, Kaiming Zhang and Xiaoming Su. And we tried to apply cryo-EM to this entire benchmark set of RNA molecules. And we were actually super excited to see that for 11 of them, we actually were able to resolve the global architectures of the RNA molecules with cryo-EM. But if you look closely at these maps, what you can see is that the map resolution is not very high. So here the map resolution ranges from about 5 to 11 angstroms. So why does that actually matter? So the cryo-EM map resolution actually specifies the level of structural detail that we get out of these maps. So I'm showing here 3 different maps of the same molecule, just at different resolutions. And the goal is to be able to use this map to figure out where every single nucleotide or every atom within your molecule sits. So if we zoom in on a 3 angstrom resolution map, you can see this is actually quite easy to do. You could imagine even manually just placing coordinates into this map like this. But once you get to something like a 10 angstrom resolution map, this is really just a fuzzy blob of density, and it becomes really challenging to figure out from this blob what does this molecule actually look like. So we wondered whether we might be able to computationally model coordinates, and as it turns out, I spent the majority of my PhD developing computational methods for model RNA and RNA protein structures and energetics, so this seemed like a really great problem for my background and skill set. So the challenge here is to take a sequence of an RNA molecule and one of these low resolution cryo-EM maps and figure out if we can predict where all of the atoms in the RNA actually sit within this map. So we developed a method called AutoDrafter to do exactly this. So what we do is we sample conformations of the RNA in a series of iterative Monte Carlo simulations. And then we score each one of these conformations with a score function that has 2 parts. So first, a statistical energy function, which scores how physically reasonable the conformation is. And then second, we have a score term that rewards agreement with the experimental density. So hopefully in the end, we end up with conformations that are both physically reasonable and agree well with the experimental data. So now that we have this method, we want to test how well does it actually work. And so to do this, we went to high-resolution crystal structures of RNA molecules that had previously been solved, and we simulated from those crystal structures low-resolution cryo-EM maps. And then we used AutoDrafter to build models into those maps, and I'm showing here the models for 8 different systems. And then when we compare those models to the high-resolution crystal structures, you can see there's actually quite good agreement, suggesting indeed this method actually is quite accurate. The really exciting thing here is that actually we realized that we can really reliably predict the accuracy of these models before we know the true answer. So we're looking here at correlation between our predicted accuracy and the actual RMSD accuracy. And this predicted accuracy we can get directly from the models that we build. And so what that means is we can actually use the models that we built to determine the extent to which we should actually be interpreting these models. So now that we have this technique, we wanted to go back to these 11 low-resolution cryo-EM maps that we solved, where we don't actually have high-resolution coordinates, and see whether we can now use AutoDrafter to build models into those maps. So we are excited to see for all 11 cases, we were able to build quite reliable structures into these maps. And I want to emphasize this entire process for all 11 of these structures, including all of the methods development, only took a few months. So there's a pretty dramatic acceleration in being able to resolve RNA structures. But I do also want to highlight that these are not atomic resolution models. So we're not saying precisely where every single atom in these molecules actually sits. So the estimated accuracy here is about 3 to 6 angstroms RMSD. And so that tells us the global architecture of the RNA molecule, but not where every single atom actually is. So are these models actually useful if that's all we get from it? So I'll give you just a few examples of things that we can learn from these models. So we're able to look at 2 very different RNA sequences and see that actually they're extremely structurally homologous. So they fold up into almost the exact same 3-dimensional structure despite having totally different sequences. So we're able to really robustly assess structural homology. We can also really robustly observe conformational changes upon substrate binding. So here we're looking at a ribozyme before and after it binds to its substrate. We can see exactly where the substrate binds and the global conformational changes that need to occur for that binding to happen. So I mentioned before that RNA molecules, they don't just act alone, but they also bind to proteins to form these RNA protein complexes. And this is how a lot of these RNA molecules actually function. So we extended this method to be able to model RNA coordinates into large cryo-EM maps of RNA protein complexes. This approach is called Drafter. And during my postdoc, we used this approach to resolve a mystery. So we were interested in this protein called ISRB, which is an ancestor of Cas9, the bacterial defense protein, which has been widely adopted for genome editing applications. And the really striking thing that you'll notice here is that ISRB is tiny compared to Cas9. And so we wondered, how is it that ISRB with such a substantially reduced? So to address this, I collaborated with a really fantastic structural biologist, Seiichi Hirano, and we used this combination of cryo-EM and computational modeling to resolve the architecture of ISRB with its guide RNA, with its omega RNA, and this on the right side here. And when we compare it to the structure of Cas9, what you can see is that this entire domain here has actually been replaced by an intricate three-dimensional pseudoknotted RNA fold in the case of ISRB. So actually the RNA is taking the place of the protein, and that's how it's able to carry out its function still. We've used this approach to characterize lots of different RNA molecules, riboswitches, ribozymes, and different kinds of RNA protein complexes. So now that brings me to the second part of my talk, moving away from the molecular scale and more towards the subcellular scale. And so I became really intrigued by this question of how is the inside of a cell organized? So a typical human cell contains about 5 to 10 billion protein molecules, which is just a massive amount if you think about the fact that specific collections of those molecules need to come together in time and space to carry out all of the processes that keep you alive. So of course, a cell is organized by membrane-bound organelles, so things like the nucleus and mitochondria. But it turns out that cells are also filled with these membraneless structures called condensates. And so these are membraneless assemblies that concentrate specific collections of proteins and or nucleic acids. And this is an animation that I like to show to highlight the difference between a membrane-bound organelle on the left and a condensate on the right. So in the case of a membrane-bound organelle, you have a membrane that's physically enclosing the molecules inside. In the case of a condensate, you don't have that membrane, and so that means there has to be something intrinsic to the molecules themselves that's causing them to self-associate. Okay, so just so we're all on the same page here, the terminology in the condensate field is a little bit of a mess. These things go by granules, droplets, bodies, same things. These condensates can form through multiple different mechanisms, and they can have different material properties. I won't talk much about the material properties today. They include very well-known examples, such as the nucleolus, stress granules, nuclear speckles. They show up as these puncta in fluorescence microscopy images. And they can carry out a number of different functions. So they can act to sequester molecules away from the rest of the cellular environment. They can act as organizational hubs to bring specific collections of molecules together in the same place to carry out a particular process. And they can also act as reaction crucibles to help speed up chemical reactions. They're involved in a very wide array of cellular processes. Honestly, almost any process you can think about, there's probably a condensate involved. So things like autophagy, carbon fixation, innate immune signaling, RNA degradation. And condensates they seem to go awry in many diseases, especially neurodegenerative diseases and cancer. So because these structures are so important, we really want to understand what types of proteins form condensates. So multivalency, very broadly, promotes condensate formation. So that means if you have a single molecule that has multiple sites where it can interact with another copy of that molecule. It turns out that intrinsically disordered proteins, or proteins that lack a well-defined three-dimensional conformation, especially tend to be multivalent. You can kind of think of individual amino acids in these molecules basically as interaction sites. And so you can super easily get multivalency from these disordered proteins. If you crack open endogenous condensates in human cells and just look at the proteins that are inside of them, the vast majority of them actually contain disordered regions. And I just also want to highlight that disorder is not a rare phenomenon. Depending how you count, about half of the human proteome is actually disordered. Okay, so disordered proteins, they tend to show up a lot in condensates, but not all intrinsically disordered proteins form. What are the sequence features of intrinsically disordered proteins that actually drive condensate formation? So if we enable us to make predictions for new sequences and maybe even design totally new sequences. So the challenge here is if we have two different sequences, one on the left has a low propensity to form condensates, one on the right has a high propensity to form condensates. What is it that's actually different about these two sequences that's causing this difference in condensate formation? Is it specific amino acid types, particular sequences, hydrophobicity, charge, amino acid patterning, or maybe something totally else? So before we dive into each of these potential possibilities, I want to back up and consider more broadly some parameters that can impact condensate formation. So it turns out that condensate formation is extremely concentration dependent. So for most proteins, as you increase protein concentration, you will increase your propensity to form condensates. Additionally, the environment that you're in can have a huge impact on whether or not you're forming condensates. So that means whether you're performing experiments in vitro in a test tube or in a cell, and even within a cell, if you're looking in the nucleus versus in the cytoplasm, these can have very different impacts on whether a sequence will form condensates or not. Okay, and then finally, what I'm really interested in is what about the sequence is actually driving condensate formation. And so this is maybe totally obvious, but if you want to understand what about sequence is driving condensate formation, we need to remember to control for these other parameters. So just keep this in mind as we go forward. Okay, so returning to this question, what sequence features of disordered proteins govern condensate formation in cells? I'm just highlighting here a few of the papers over the past several years that have tried to address this question. So there's been a ton of fantastic work that has gone into this. And just to give an overview of how these studies typically work, people will typically take a single model protein and they'll test up to tens of different sequence variants and looking especially for variants that disrupt condensate formation. Variants conclude something about what's actually important for driving condensate formation. So in this case, maybe arginine residues are really important for driving condensate formation. This is then, repeated in a different study for a different protein, testing different variants and, maybe in totally different experimental conditions. And in this case, maybe the authors conclude aromatic residues are really important for driving condensate formation. And so to summarize a ton of work, depending on the starting protein and which variants are characterized, pretty much every amino acid type, like almost every feature that you can think of these proteins has been shown in some context to drive condensate formation. And so this becomes challenging then to get sort of systematic rules and understand generalizable principles that are driving condensate formation across different contexts. So we wondered whether there's a way to more systematically assess sequence features that promote condensate formation in cells. So there are really two classes of existing methods for characterizing condensate formation. So the first class of methods characterize condensate formation in vitro. And by in vitro, I mean in a test tube. And these are very powerful methods. They provide information about intrinsic protein biophysics, but they don't necessarily tell us how a protein will behave in a biologically relevant environment. And so I'm really interested in understanding what happens in the cellular context. So to do that, we like to make measurements inside of cells. This has the advantage that it tells you something about what happens in a biologically relevant environment, but existing methods to measure condensate formation in cells measure protein sequences one by one. And so if you're measuring sequences one by one, you can see why all those previous studies were focusing on one protein and looking just at a few variants. This really limits scalability. So we ideally need a new approach, an approach that makes measurements not just for one protein, but for lots of proteins, and can make measurements for lots of rationally designed variants of these proteins, and that makes measurements all in the same experimental conditions. So we developed an approach called condensates that enables us to do exactly this. So the first step is to create a sequences. So we can test up to about 5,000 design experiment. We design DNA sequences that encode the proteins that we want to test, and we have them synthesized in a pooled format. So we end up with a single tube of DNA that encodes all of the proteins that we're interested in. We then clone these proteins these DNA sequences encoding the proteins into an expression vector that contains a few essential components. So first, an inducible promoter, which enables us to control the timing and level of expression of these proteins. Then for each test protein, we have a barcode sequence, which is just a unique DNA sequence, which will uniquely identify each test protein. We have the test protein itself. We have a nuclear localization signal. And so this is going to enable us to study condensate formation, at least for now, specifically in the nuclear environment. Remember, I said there's differences between the nucleus and the cytoplasm. So we want to confine our measurements to one particular environment initially. We then have a fluorescent protein, which enables us to visualize the molecules in cells. And then optionally, we include an oligomerization domain, which enables us to control the valence of these constructs. So then we take this pool of plasmids and we put them into human cells. We've used HeLa cells for the majority of the experiments I'm going to show here. And we put them in cells so that every cell expresses just one of these protein sequences. So then we have a pool of cells. Every cell is expressing a different protein sequence. And then we can image this entire pool of cells. I'm showing an example image here. You see lots of cells showing up. And every cell looks kind of different. In some cells, you see a diffuse localization pattern, no condensates forming. In other cells, you see these condensates really robustly forming. And then the challenge is, so they all look different because they're all expressing different protein sequences. And so if we want this data to actually be useful to us, we need to figure out which protein each cell is expressing. Okay, and so to do this, after we image these cells live, we then fix them, we keep them in that same dish, and then we detect the barcode sequences directly in these intact fixed cells through in situ sequencing by synthesis. So we're going to synthesize a complementary strand of DNA to this barcode sequence using fluorescently labeled nucleotides, where every nucleotide has a different color. We're going to incorporate one nucleotide at a time and then image after each nucleotide incorporation. So I'm showing the live cell image that we took here. And then after the first nucleotide incorporation, this is what the image would look like. So you see all these colorful spots showing up. And those spots will tell us the identity of the nucleotide at the first position of the barcode. So you see these blue spots in the center here. That tells us there's a C at the first position of that barcode. We go to the second nucleotide incorporation. You see they changed to red. That tells us there's an A at the second position of the barcode. They turn pink at the third position. That tells us there's a T at the third position of the barcode. So now we know that cell contains a CAT barcode. We can repeat this for however many nucleotides we have in our barcode. And then we know for every cell which barcode it expresses. And then we can go back to our original library that we designed, and we can look up what protein sequence corresponds to that barcode sequence. So we then have a bunch of automated image analysis software that will then sort all of these cells based on the proteins that they're expressing, and then will automatically detect and quantify lots of different properties about these condensates. Okay, so remember I said that we need to control for concentration and environment if we're really interested in looking at sequence. So this method nicely controls for the environment. So we're looking always in cells and specifically within the nucleus of cells. Does it control for concentration though? So actually we're going to quantify condensate formation as a function of concentration, and we can do this in one of two different ways. So either we can induce protein expression and image over time so that we can observe the first concentration at which we start forming condensates. So we'll get basically a threshold concentration for every single protein sequence. Or what we can do is we can look across our entire population of cells, and we can look just at the cells within a defined concentration range. And then within that defined concentration range, we can compute the fraction of cells that contain condensates. These are obviously highly related metrics, but this one is a little bit more scalable than the first one. And so this is the metric I'll use. Okay, so indeed, we have a method that controls for concentration and environment and enables us now to look specifically at the sequence determinants of condensate formation. So now that we have this method, does it actually work? So we first tested this with an initial small sequence library. We looked at a library of 99 short protein sequences and just performed two different replicates, looked at the fraction of cells that formed condensates in each replicate. You can see there's very good agreement, suggesting at least this is technically reproducible. What we did then is we compared these pooled experiments to arrayed experiments, so where we put one single sequence in every single well so that we know exactly which protein sequence we're looking at in every well, and we don't need to do the in situ sequencing step. And so I'm showing some example images looking at the arrayed images from the arrayed experiments on the top and then images from the pooled experiments on the bottom. And you can see they agree really well, suggesting that we are actually accurately detecting the barcodes in these experiments. So these experiments do seem to technically work. So now we can actually go back to the question that we're really interested in. Across a wide range of disordered protein sequence contexts, what are the sequence features that have the biggest impacts on condensate formation? So to begin to address this question, we actually went back to all that literature that I showed you at the beginning of the talk, and we took all of those studies basically as hypotheses that we can now test, not just in one system, but across a very wide range of different systems. So we designed then a library of about 14,000 disordered test protein sequences. And this library includes about 1,600 fragments of disordered regions of natural proteins. And this part of the library was basically just selected to maximize diversity of the final set of sequences. So we can cover as much of sequence space as possible within our library. We then have about 9,300 variants of a set of about 80 starting sequences where we have basically systematically made mutations to test every single one of the hypotheses from all of those papers on the previous slide. So where we're adding and removing each instance of every amino acid, we're changing net charge, hydrophobicity, et cetera. 600 scrambled variants of a set of starting sequences. And so these are variants where we're changing the ordering of the amino acids without actually changing the composition of the amino acids. So we ended up with tons and tons of data from these experiments, more than I'll be able to tell you about in this talk today. But I do want to highlight that actually nearly all of the sequence features that we tested did have some effect on nuclear condensate formation. So this does seem to be a pretty complicated process that's going on here. But to give a sense of what are the strongest and most consistent effects on nuclear condensate formation, we look here at the normalized effect per single amino acid substitution. So I have the type of mutation on the x-axis, and then the normalized effect on condensate formation on the y-axis, where a positive effect increases condensate formation and a negative effect decreases condensate formation. And you can see that the biggest increases come from adding aromatic residues, adding positively charged residues, or removing negatively charged residues. And then if we look at what the biggest decreases come from, we see the exact opposite effects. So we see the biggest decreases come from removing aromatic residues, removing positively charged residues, or adding negatively charged residues. And we can look at this in a different way. If we take all of our 14,000 sequences and we bin them by the number of aromatic residues they contain on the x-axis and the net charge per residue on the y-axis, and then for every bin, we compute the average fraction of those sequences that are forming condensates, where yellow indicates all of them are forming condensates, black, none of them are forming condensates. You can see as we increase the number of aromatic residues, we increase propensity to form condensates. And as we increase net charge per residue, we also increase propensity to form condensates, and we can kind of compensate for one. So the number of aromatic residues makes a ton of sense and is super consistent with all of the previous studies that I showed you on the previous slide. This has been found numerous times that aromatic residues are important for driving condensate formation. But net charge was a little bit more surprising. So if we were just looking in a test tube, you would imagine that high net positive charge probably would not actually be driving condensate formation. It would probably drive electrostatic repulsion. So how could it be actually promoting condensate formation? But remember that we're performing these experiments inside of cells. And so, of course, these proteins can interact with all of the other things that are actually inside of cells. And so we wanted to get some sense of whether those other factors are actually playing some role in driving condensate formation. So we first just wanted to ask a very simple question. Cells are actually filled with lots of endogenous condensates. Are these test protein sequences that we're putting into the cells actually just co-localizing with endogenous condensates? Are they just going and joining existing condensates? And so to test this, we can assess directly co-localization of these test protein sequences, at least one by one, via antibody staining. So we can have our GFP fused test protein. We can have a marker for something like the nucleolus, look at the overlay, and see here we're getting perfect co-localization with the nucleolus. This is a sequence that's just going and localizing to the nucleolus. So we used this approach for a set of 59 sequences. About 20% of them co-localized with endogenous nuclear condensates, nuclear speckles, the nucleolus, and chromatin. But these 59 sequences are not really enough for us to derive sequence trends to understand what are the principles driving co-localization with endogenous nuclear condensates. And for technical reasons that I won't get into, it's relatively non-trivial to actually scale this approach to 14,000 sequences. So what we really wanted is a way to go directly from these images of the GFP-fused test proteins that we already have for our entire library and just use that to assess colocalization. And so here what we did is we thought we could focus maybe on morphologically distinct structures, so things that are really readily identifiable, so nucleolus and chromatin, and then maybe be able to identify that localization directly from these from these test protein images. So we developed a classifier that does exactly this. It takes an input of a live cell expressing a GFP tagged test protein, a condensate classification. So out sequence localizes to the nucleolus, to chromatin, forms other kinds of condensates or doesn't form condensates at all. This is quite an accurate classifier. You can see some of the example images on the right, nucleolus classifying images on the top, and chromatin localizing images on the bottom. And so now that we have this approach, we can actually apply this to our entire library of 14,000 sequences. And what we find is that about 12% of our sequences co-localize with the nucleolus, about 4% co-localize with chromatin. And now we have enough statistics to actually try to understand what are the sequence features that are driving this co-localization. And so what we saw is that the biggest feature, the feature that had the biggest effect was net charge. So here we're looking at net charge on the y-axis. You can see for the nucleolar sequences and the chromatin localizing sequences, they tend to have much higher net charge than sequences that form other kinds of condensates or don't form condensates at all. And of course, this kind of makes sense. These sequences with higher net charge are likely associating with negatively charged nucleic acids. Okay, so then we could ask more broadly, actually, what are the sequence features that are important for self-interaction versus interaction with other cellular factors? So to get more broadly at this impact of the rest of the cellular environment. So again, CondensSeq does not directly tell us about interactions between test proteins and endogenous cellular factors. So we need another approach. So we turned to this computational method called Finch's, which was developed very recently in Alex Holhouse's lab at Washington University in St. Louis. And this method really reliably predicts interaction strength between disordered regions. So what we can do with this approach then is we can take our entire library of 14,000 test protein sequences, and we can consider potential interactions across the entire human proteome. So what we did is we took all of across the entire human proteome, there's about 4,000 endogenous a certain length threshold. And then we can ask for every test protein, we can compute the self-interaction or homotypic interaction strength. And then we can also compute the interaction strength between one of these test proteins and every single one of these endogenous disordered regions. And then we can ask for each test protein, what is more favorable? The homotypic interactions, so self-interactions, or heterotypic interactions, interactions with other cellular factors. And then we can take all of our condensate forming sequences that we identified with CondensSeq, and we can split them into two groups. So one where the homotypic interactions are favored, and one where heterotypic interactions are favored. And then we can ask what's actually different about the sequences in these two groups to get at what is driving homotypic interactions versus heterotypic interactions. So I'm showing here a dot plot of sequence features that differ between the two groups. Haven't labeled out everything here because this is a little bit overwhelming. The red dots indicate that the sequence features are higher when homotypic interactions are favored and blue when heterotypic interactions are favored. And just to zoom in on a couple features, we see that the number of aromatic residues tends to be higher for condensate forming sequences where homotypic interactions are favored. So suggesting that aromatic residues are really driving condensate formation through homotypic interactions. On the other hand, net charge tends to be higher for condensate forming sequences where heterotypic interactions are favored, suggesting that net charge is driving condensate formation through heterotypic interactions. Okay, finally, we can take this analysis even one step further and actually ask, how might interactions with endogenous cellular factors actually tune the sequence features that drive condensate formation? So if we return to our Finch's analysis, this analysis actually gives us a predicted interaction profile for each test protein. So for every one of these test proteins, we have its predicted interaction strength with every endogenous IDR, human IDR. And then what we can do is we can group these test proteins by the patterns of predicted interaction strengths with endogenous human IDRs. So for example, we might have group 1, which contains test protein 1 and 2, which might interact really strongly with human protein 1 and 4. You might have another group that contains test protein 5 and 6 that interact really strongly with human protein 3 and 6. So we can then take all of these test protein sequences and we grouped them into 10 distinct groups where all of the sequences within each one of those groups make similar sets of predicted interactions. And then for each group, we can ask what's different about sequences that form condensates versus do not form condensates. And again, this is a gigantic, complicated dot plot. So the red dots indicate sequence features that are higher for sequences that form condensates, blue lower for sequences that form condensates. And so each row here is a different sequence feature. And so I just want to highlight a couple of things that can happen here. So here we're looking at hydrophobicity and the effect across different groups of test protein sequences. And you can see that actually in some cases, hydrophobicity is higher for sequences that form condensates, and in other cases, it's actually lower for sequences that are forming condensates. So basically saying, depending on what kinds of interactions you might be making with other things in the cell, hydrophobicity can either drive or inhibit condensate formation. Net charge, what we can see, we don't see sort of the opposite effects. We see that it's always either higher net positive charge for sequences that form condensates or no effect at all. But what we do see is that you don't always need higher net charge to drive condensate formation. So you can see there are some groups where it's there's a very little difference in net charge between groups of sequences, between the sequences that form versus don't form condensates. And in other cases, it's completely unimportant altogether. So what does this analysis tell us? It tells us that the relative importance of sequence features for promoting condensate formation actually varies depending on these predicted interaction profiles. So basically all of this to say that the entire cellular environment is actually extremely important for determining what is driving condensate formation. Okay, so just to summarize this part of the talk, we found that the number of aromatic residues and net charge per residue were some of the strongest and most consistent drivers of condensate formation. But we are operating through distinct mechanisms. So aromatic residues seem to be especially important for driving condensate formation through homotypic interactions, whereas high net positive charge seems to be important for driving condensate formation through heterotypic interactions. And we see that interactions with endogenous cellular factors are really important. So if you want to understand what's going on inside of a cell, you should probably actually make some measurements in the cellular environment. So we've used CondensSeq to do a bunch of additional things that I haven't talked about here. We've used it to map the impact of protein valence on condensate formation, to assess the roles of amino acid patterning, to identify sequence features that actually drive nuclear concentration buffering, and to look at sequence features that control condensate size and number. But many, many questions still remain. So we're very interested in how sequence controls condensate dynamics. How are these condensates actually regulated in the cellular environment? We know that most of these condensates contain RNA molecules. How is the sequence of the RNA actually contributing to condensate formation? Cells contain numerous different condensates, and those condensates maintain distinct compositions. How is that composition actually controlled? And finally, we're really interested in understanding what are actually the functional implications of forming these condensates for different specific systems. Okay, so that leads me to just give a short outline of what we're thinking about in my own lab. So returning to this number that I threw out at the beginning of the talk, that there's about 8 billion known protein and RNA sequences. And I suggested that we might want to try to understand what all of those sequences do and how they do it. And I think we made a little bit of progress towards getting there. We can more efficiently and more quickly resolve RNA structures with cryo-EM and computational modeling. And we can characterize sequence condensate relationships for, not just tens of sequences, but thousands of sequences using condenseq. But really, we're nowhere close to characterize billions of sequences right now. And actually, the problem is way worse than just billions of sequences, right? If we just think about the total amino acid proteins, it's a really small protein. There's 20 to the 100th possible sequences. If we consider just the mass of DNA that encodes 20 to the 100th protein, so just if we have one molecule per sequence, which is basically the minimal thing you need to actually be able to characterize these sequences experimentally, that's 6.4 times 10 to the 83rd times the mass of the Earth. So it's actually physically impossible for us to characterize sequences at this scale. And so what we're really interested in doing is figuring out whether we can actually make predictions. If we want to get to this kind of scale, we need to be able to accurately predict sequences. And so we want to be able to predict organization and function from primary protein and RNA sequences. And so in my lab, we do this through a combination of developing high throughput methods and making measurements for lots and lots of sequences, and then using and developing computational methods to learn from those measurements and extrapolate to sequences that we have not tested experimentally. So we're trying to get towards predictive models of condensates, RNA structure, and RNA protein interactions that can be harnessed both for biological discovery and for design. So my lab at UCLA is about half wet lab, half dry lab. And just to give you a tiny sense of some of the dry lab stuff that we've started doing in the lab, we have this huge data set of sequences where we have measurements of condensate formation. Can we now start actually predicting condensate formation directly from IDR sequences? So I was super lucky, actually, on the second day, the day after I started at UCLA, I had a team of undergrads, Mia Lesser and Jaden Haw, come and join and start trying to make predictions from our experiments. And then this quarter, we've had a really fantastic bioinformatics rotation student, Claire Shea, come and start trying to work on this problem. And I just want to highlight for a couple seconds some of the stuff that Claire has been doing. Our protein sequences and encoding those protein sequences in just like the simplest way possible. So basically taking our sequences and encoding them as a length 20 vector that tells us the fractions of each amino acid. So this is just considering amino acid composition. And she's built a super simple deep learning model, a multi-layer perceptron, which will then take this input and predict whether a sequence forms condensates or not. And from this super simple model, she's actually already been able to get to about 82% accuracy, which is actually pretty remarkable from such a super simple model. And we can already see she's trying to figure out like what is the model actually learning. And by doing some different feature attribution approaches, she's actually figured out that the model is already learning features that we found to be important in our experiments. So we can see that charged residues and aromatic residues seem to be the biggest things that are actually driving prediction in this model. Again, the biggest factors that we saw actually impacting condensate formation. We have tons more to do here. And so I want to highlight, if this sounds interesting to you, definitely please reach out to me. And with that, just like to acknowledge a ton of people, this covered work that I started in graduate school, did in my postdoc. So huge thanks to my graduate advisor, Riju Das, my postdoc advisors, Aviv Regev and Feng Zhang, some really amazing teams I've gotten to work with along the way, especially Hua Chu for work with on the RNA structure determination. A huge thanks to all of my lab members here who've been brave enough to join a totally new lab and are helping me like literally set things up as we speak. And thank you all for having me here today, and happy to take any questions.